All right. Thanks everybody for joining us tonight. I know I got several people online as well as here in the room. So we're so excited to have our second medical scholar seminar of the semester. This is our series that comes from the medical school, and it's joint with our translational biology, medicine, health graduate program, bringing together our medical students and our graduate students, thinking about how can we make sure that the research that is going on is truly informed by what's happening at the level of the patient. And so when we go about soliciting people to nominate people to come in for this particular series, what we're looking for are inquiry-driven scholars who are really actively engaging in patient care and using that to inform the research that they're doing, with the idea being that, you know, some of you may go out and do this or be interacting with physicians actively being informed by what they're learning every day in the clinic. And so we're excited tonight to have another member of this medical scholar community come. And I want to sort of prelude, be thinking about, for those of you in the audience, if you know of a physician who might fit this model, be thinking about who you might want to nominate, who might you want to work with to bring here next year to hear in this series. So we'll be calling for those nominations in a little while. But I'm going to turn it over to one of our FBRI faculty who runs a Center for Health Behaviors and Exercise at FBRI. Dr. Jin Yan is going to introduce our speaker. Thanks so much.
Thank you. Yeah, my name is Zhen Yan. I'm the director for the Center for Exercise Medicine Research at the Fralin Biomedical Research Institute. Welcome to the Medical Scholar Seminar. And today, it is my extreme great pleasure to introduce our speaker today, and Dr. William Cross, Bill Cross. He is a longtime friend, colleague, and competitor, I could call it, in the field, but he's my role model. And we were trained by one individual who once was the dean of the Duke Medical School, the president of Gladstone Institute, Dr. Sandy Williams. I owe him a tremendous amount of gratitude. And Bill, I've known him for years, but I just learned I've never paid attention because I never get a chance to pay attention to his CV. And it's very exhausting to read his CV. But he got a bachelor's degree from Harvard in a field anybody can guess, in some area. Can you guess which area? Astronomy and astrophysics. Wow, yeah, this put us in perspective, how tiny we are, right? But he's a really colleague, a pioneer in the area of physical activity for years. He studied. Reading his CV made me extremely humbled. And he published 530 some papers, and all high profile journals. And I won't be able to go through that. 53 book chapters. Every 10 papers, he write a book chapter. And he has a H-index of 128. That's a double of the amount of I have. This is really humbling. And he is the former president of the American College of Sports Medicine. And he's currently running this MotorPack, which is a consortium in the country for studying the transducer of physical activity, which I was honored enough to be part of the initial 13 members who were invited to NIH at the round the table discussion to start discussion what's the next step in exercise research. Bill and I noticed that he recently get into this field of exercise mimetics, which is really in line with his background in medicine. When I was at Duke, we were in the same division, and but now you have titles in 5, 6, 7 different institutions at Duke and outside Duke. He was once the director of the Center for Living, which is a very important area of research. Unfortunately, they disconnected that or discontinued that center. Yeah. Anyway, So without further ado, and Bill Cross.
Thank you, Zen, for that. You can hear me, right? Way back there in the back? You can hear, okay, good. Yeah, I'm honored to be here to give this lecture. I really appreciate the sentiments and the context in which I've been asked to do this. And I'm hoping that you will find some relationship. It's been nice to meet old friends and meet new friends. And my first visit to Roanoke, which is a charming place to live and I'm sure to work. So it takes a lot of support to do work like this. I just want to acknowledge, and I will again, I'm a member of the Duke Molecular Physiology Institute, Duke Heart, which is the cardiovascular center at Duke. And then I've had support through the NIH, through National Heart, Lung, and Blood, NIDDK, and I'll talk about MotorPack here in a minute. So between 1998 and 2013, I had the pleasure of doing 3 5-year funded clinical trials, randomized, of exercise exposure, where cardiometabolic health is the outcome. Changes in lipids, glucose control, blood pressure, body mass distribution, if you will, and cardiorespiratory fitness as the outcomes. I'm going to talk about STRIDE 1 first, and I'm going to talk about STRIDE prediabetes second. I won't talk about aerobic and resistance training in combination. The whole point of these studies, and the whole point of my career in this area of research, is to understand how much, what kind, and what intensity results in what outcomes, okay? And whether or not we can prescribe exercise in a, I'm not going to say personalized way, but in a precision way, depending upon what you have to treat, which exercise exposure is best, okay? So we'll address this first. I'll address the clinical studies underlying the work, and then I'll talk about how we've been exploring mechanisms. How does this actually happen in the body, which is a complicated process, which organs are involved, which signaling molecules, but we'll focus on skeletal muscle as the target here. We'll focus on insulin sensitivity as the outcome, the ability of muscle to uptake glucose in response to a dose of insulin or in response to a load. If anyone went to the reception, if anyone ate a brownie or cookie, most of that is being uptaken by your skeletal muscle as we speak, okay? Postprandial glucose uptake. So in STRIDE 1, the characteristics of the individuals were middle-aged, overweight or class 1 obesity. They had to have abnormal lipids, either HDL or LDL cholesterol. They could not have frank diabetes. In 1998, the diagnosis of diabetes was 140 milligrams percent. It's now 126. They had to have blood pressure not high enough that exercise or lifestyle couldn't be the first intervention required. This has also changed in the ensuing 20 years. And if they're a woman, postmenopausal. And been postmenopausal such as time that if they were on hormone replacement therapy, this is important because this was still being given as a preventive therapy in 1998. Postmenopausal women, it affects lipids, it affects insulin sensitivity in a favorable way. So if they were on hormone replacement therapy for at least 6 months. So this was the prescription in lay terms. We asked people if they were randomized to do brisk walking equivalent 13 miles per week, and it took them on average 170 minutes per week to do that. This was divided among several sessions. Jogging, 13 miles per week, 120 minutes took them. And jogging, 22 minutes, 170 minutes. Now, these have pertinent. They weren't designed this way because all these data weren't available at the time. But we call this the physical activity guidelines group. Because if you know what the physical activity guidelines for Americans is, it's brisk walking 150 minutes per week. Okay? This we call the fitness guideline because for years, the guidelines were exercise at vigorous intensity 3 times per week for about 30 minutes per session. So that fits there. And then the jogging week, we call this the Harvard Alumni Study Group because this is the exposure that reduced to the greatest extent cardiovascular events and mortality in the Harvard Alumni Study, Paffenbarger study. And then we had an inactive group. Now, I used to call this a control group, but in a control group, what do you want? You want stability, okay? Americans with the previous demographic are not stable over the course of 6 months, and you'll see this in a minute. So now I just call them inactive. By the way, there was no dietary prescription in any of these groups. We monitored diet and didn't see any changes in diet over the course of the intervention. So none of these effects are due to diet. Okay, these are the actual details for an exercise physiologist, right? They were exercising at 40 to 55% of their peak VO2 or 65 to 85% of their peak VO2. And we call this low moderate, low amount, moderate intensity, low amount vigorous, high amount vigorous. And we're matched on energy expenditure when we're matching. So when in these two groups, you compare these two groups, this is important. You compare these two groups, you're looking at an intensity effect because they have the same energy expenditure per week, the same amount. So any differences are due to the vigorous versus the moderate intensity. You compare these two groups, you're looking at an amount effect. It's the same intensity, but different amounts. This was the key to the whole design of the study to allow us to understand how much, how intense, right? And it's a dose response study. The best way to show mechanism in any trial is dose response. When people are developing drugs, what do first? They do a safety check. And then they do a dose response. The phase 2 study is a dose response study, okay? It's the same thing with lifestyle. If you want to show effect, the best thing is to have a dose response thing. Now, we could look at frequency, but we weren't designed to do that. This was our exposure, 3-month ramp up to avoid musculoskeletal injury, going from doing nothing to something very vigorous. And then 6 months of steady state training. And then we had 2 weeks of detraining to look at withdrawal exposure to see what happens, what reverses, okay? That's another way of showing mechanism, by the way. But I'm not going to talk about the detraining time points today. Okay, so this was what we predicted in the grant when we wrote the grant. We predicted a dose response in changes in cardiorespiratory fitness. So the inactive group had no significant change in cardiorespiratory fitness. They're already unfit, they stayed unfit. Okay, then there was a 6% increase, 7% increase in the brisk walking group at low amount. And vigorous had a more robust response in cardiorespiratory fitness. This is known. This was known. We predicted this would happen based upon what we knew. This was our hypothesis. We'd see a dose response by amount, right? The more you do of that vigorous, the greater improvement you get. Athletes know this. They're constantly exercising, right? To keep that highest level of fitness. So predicted, et cetera. But you're looking at the bars and you're not looking at the error bars. This is an error we always make in looking at data. What does the error bars tell you? Variability in response, right? So what does that variability look like? Looks like this. This is the control group, okay? Roughly, you add all these up, you get roughly no change, right? This is the mild group. This is the low amount, moderate intensity. So a less of a response, but a response. This is the vigorous, low amount, and this is the high vigorous. So if you're in the high vigorous, this is within the error of the measurement. 10% is the error of the measurement. That's what these solid lines are, right? So if you're in the error of the measurement, you don't know exactly if you had a response. But if you're going to take a vigorous intensity exercise, don't you want to know if you're this person or this person? Right? This is the whole thing about personalized medicine. Can we develop markers to identify if you're going to be this person or that person? I'm not going to talk about that today, but that's the point. It's the same thing, medical students with drugs. They won't tell you this, but they're unresponders to drugs, right? Or they're poor responders to drugs, or they're adverse responders to drugs, or they're super responders to drugs who need less of a dose. And some of this changes with age and body organ function. Okay, so this is percent change in body mass, percent change in body mass, okay? So this is why I call it an inactive group, because in 6 months, those who were inactive group gained 1% body mass. Okay, so for a 100 kilogram man, that's 1 kilogram. That's 2.2 pounds. That's 4.4 pounds in a year. And when I first started showing these data, everyone said, no, 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 no. And I said, I'm from North Carolina. And you come down to our airport and look at who's getting out the airplane. And you'll change your mind, because this is exactly the human condition right now. Okay? Any exercise ameliorated that, any of them. And actually, the more you did of the high vigorous, the more energy you expended, the more decrease in weight. Now, I'm going to dispel a dogma. It's not a dogma in the scientific field, it's a dogma in the lay field. Exercise causes weight loss. You do all this work and you lose 4 pounds? Is that what people are looking for? No, no. Exercise alone, in absence of diet, does not cause significant weight loss. It causes weight distribution a little bit, and that might help some of your cardiometabolic parameters, but it doesn't cause weight loss by itself. You need the diet to cause the weight loss and the exercise to maintain your weight. That's the lesson here, okay? So far, no big surprises. This was the first big surprise. This is the change in insulin sensitivity index with the intravenous glucose tolerance test. What we do is we give a bolus of IV glucose. It's basically a Mars bar in liquid form. We give a bolus. Their glucose goes up. The pancreas responds by increasing insulin, and the glucose starts to come down. Then we give a little dose of insulin, IV, and it brings the glucose down more dramatically. That second response is the insulin sensitivity of the muscle. It's basically a muscle insulin sensitivity test. Okay? We're interested in muscle and its effect on body habitus. We're interested in exercise effect mediated through muscle on insulin sensitivity. So we use this test in an index, actually measuring the muscle response to the exercise program. I won't go into a lot more of the science and detail here, but look at this. The brisk walking group had the most robust response. And if you did the same amount at vigorous, you had half of the effect. And it took 50% more kilocalories per week for the vigorous group to get so what the heck's going on there? Right? What the heck's going on? How can that be? Well, all the exercise guys in our fields then say, this can't be. More has got to be better. Hard has got to be better, but it's not. And if we did metabolic syndrome index, which is a combination, won't go into details, of fasting glucose, waist circumference, mean blood pressure, HDL cholesterol, and triglycerides. We form a Z-score with that. Its response to these programs was exactly the insulin sensitivity response. So it's not just the insulin thing, right? Something's going on here. It's possible that at lower intensity exercise, we know this for a fact, you tend to burn more fats than glucose. Your RQ is lower, okay? As you increase the intensity, your RQ gets higher, closer to 1, where you're burning glucose over fats. It could be that the lower intensity exercise is consuming fat in the muscle, decreasing the negative effect of fat in muscle has on insulin sensitivity. And that's why we're seeing this. We haven't proven it, but we have some other evidence to support that hypothesis. But this is the first big surprise coming out of STRIDE. And everyone said it can't be. Well, we looked at another marker that's insulin sensitivity, is serum triglycerides. That is also, that's fat sensitivity to insulin. And look at that, that looks the inverse, decrease is better. It looks at the inverse response is the insulin sensitivity. So we think this is a real biological finding. This isn't an artifact of our testing. Okay, second big surprise. Remember, half of the women were on hormone replacement therapy and half were not. So that gave us the opportunity to say, does hormone replacement therapy interact with exercise to affect insulin sensitivity response? So you saw this before. Here are but here in the dark bars are the men and the white bars are the women. Okay, so the women at whole look like the men. But if you break the women out by not on hormone replacement therapy and on hormone replacement therapy, you see something very different. In moderate intensity exercise, it doesn't seem to matter. But if you're on hormone replacement therapy and you do vigorous intensity exercise, you get a good response. If you're not, you get no response. Okay? So if I have a woman who's coming in insulin resistant, first of all, I'd probably give them the moderate exercise dose anyway, right? But if they're not on hormone replacement therapy, which fewer women are these days for other reasons, I'd say don't do vigorous intensity exercise. But what we think is going on with the insulin sensitivity thing is that there's there's a contrary effect happening with vigorous intensity. They're getting the thing that they get with moderate, but there's some other process going on. Is it inflammation with the vigorous intensity exercise? What is it that's kind of mediating it? Which then brings us to my next part. We're about doing mechanism. So what we do is we collected muscle biopsies at all these time points in all these people so that we have tissue to analyze. Not only looking at it with a test, an intravenous glucose tolerance test, but on a molecular basis. Can we identify intracellular processes that are mediating these responses? So just to show you that the thing I showed you for fitness is not, this is my friend and colleague, Kim Huffman, I've worked with for 20 years. She did this analysis. And so this waterfall plot, we call it, that you saw for fitness is also true for each group in the insulin sensitivity response. Okay. So we can look at variability and see if we can get predictors. So just to get back one more thing. So we had the opportunity in the third STRIDE study to retest our finding from STRIDE 1. Is moderate better than vigorous? So this time we had two high amounts, high moderate and high vigorous, and a low amount moderate to compare to our previous study. And we had oral glucose tolerance tests as the outcome. We had about 150 people in this analysis, and we had the same kind of design, okay? We're looking at same amount, brisk walking versus jogging, and now we have the higher amount of brisk walking. So these are the higher amounts, higher energy expenditures per week, different intensities, and then we can look at comparing these two groups, we can look at amount. And the comparison here was not a control group or an active group. It was the landmark finding of the diabetes prevention program. Okay, in the diabetes prevention program, people who were pre-diabetic were randomized to a lifestyle group, a metformin group, and a control group. And they developed diabetes in the control group at about 10% per year for the 4 years of the study. 40% converted to diabetes. The metformin group reduced that by 32%. The lifestyle group reduced by 64%. Okay? And that was diet to reduce body mass by 7% in 6 months and a moderate intensity exercise program. So we exposed people to that for 6 months. And we got that response in those people. And this is the gold standard. And we're comparing exercise alone. So how does my clinical practice bear on why we ask this question? Okay. I counsel on lifestyle in my clinic, in my cardiology clinic. I'm doing a prevention cardiologist. I have to counsel on smoking cessation. I have to counsel on physical activity. I have to counsel on diet, stress reduction. That's 4 things right there. You start to do that in all one clinic visit, it glazes over their eyes. Or you do like the primary care doctor who's gone out the door, they say, by the way, lose some weight and get some more exercise, right? This takes time in the clinic, right? So I'm an exercise guy. I want to focus on the exercise. So I want to know, of the diabetes prevention program effect that I told you is really dramatic, how much of it's due to the exercise alone and how much is due to adding diet to it, right? So that's why we had those groups. That's how I got funded. And here's the answer. Okay, just focus on this. This is area under the curve glucose in an oral glucose tolerance test. It's basically kind of like the same. You give oral glucose, most of it gets taken up in muscle, okay? So a reduction is good. So if the glucose doesn't go high and come back down, it goes up medium and comes back down because it's being disposed of by insulin in the muscle, right? Then that's better. So a lower thing is better. All right, so in the gold standard group with 100%, 94% of that effect without diet can be by prescribing and have people do a moderate intensity walking group. Now, is that amazing? Is that surprising? Is that clinically pertinent? Yes. Okay. And go, look at this, high vigorous. Same amount. Okay. Enough said? All right. Why are these different? Why are these different? Is something going on with the vigorous intensity exercise that is against glucose control, right? All right. So we started the Molecular Stride Project. All this work started in 1999, and my colleagues were Kim and myself, and then Eric Hoffman at Children's in DC. I understand you have a connection to Children's here, okay? So I worked with Eric Hoffman and Dustin Hiddle and Monica Hubel, who I'm still working with, okay, out of that collaboration on the molecular stride work, okay? This is the first time we did a gene expression array on our muscle, way back in 1999. And then the current phase started in 2001. I have my collaborators at Duke, University of Minnesota, CC Mon, Constantine Aliferis are doing the statistical modeling. It's causal modeling. It's not just linear modeling, it's causal modeling. How can we identify what's leading to what? You can do that in a temporal study like this, right? What comes first? And then Indiana University, Monica and Rochester, Matt Morris and Gordon Broderick. You'll see pictures of them in a minute. So our questions in molecular stride are this. What are the molecular mechanisms underlying these clinical responses? Okay, I'm going to talk about that first. Can we identify known compounds in a database already studied, okay, that target KEAT nodes and such pathways? There's a whole database of drugs that have been abandoned. but we know what their targets are, right? So they've been studying it for cancer, or they've been studying it for this, or they've been studying for that, but they know it has, molecule BDD as a target, right? If we identify BDD as a target, we could repurpose that drug, right, for benefit. And can these serve as clinical mimetics of targeted clinical outcomes with or without exercise? There are people that can't do moderate intensity exercise 5 days a week, okay? If they need insulin sensitivity response, can we identify a drug that will reproduce that? So I like this, the elephant in the room, right? He's looking at an elephant is like a big snake, da, 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 da. They're all blind. They don't know what they're seeing. It's the same thing with exercise, right? Is age a cause? Is training type a cause? What I'm just talking about? Is genetics a cause? Is sex a cause? Is hormone replacement therapy a cause, right? So dissecting the complexity of exercise, here's what we do. We have the biological variability, I just told you. We have physiologic mechanisms, okay? Different groups, different responses. We study what's called the genomic paradigm or the genetics paradigm, right? The central dogma, you heard me use the word dogma, right? This is the central dogma of molecular genetics, right? Gene leads to transcription of RNA, leads to translation of protein. So if protein is the actor, and by the way, metabolomics is under here. If protein is the actor, where along the chain is the action, is what we're trying to find out, okay? And these are target specific. These are organ specific responses. Exercise affects in insulin sensitivity, the pancreas, the liver, the muscle, but who's calling the shots? Is muscle calling the shots, right? Is muscle signaling to the pancreas? How's the pancreas getting the signal? To increase insulin sensitivity and decrease insulin output. So putting it all together is what leads to us being able to identify individuals by their potential response or groups of individuals. You'll hear the words precision medicine and personalized medicine. Let me just say, the consensus of an NIH panel last year is we should use these words specifically. Precision is targeting therapies to groups of people based upon their characteristics. Personalized medicine is targeting an individual based upon their characteristics. So in the future, we'd like to understand, can we personalize really, or can we really only do precision medicine here? Motor path was alluded to by Zen. We will have, when we're done, 1,541 sedentary adults undergoing 12 weeks exercise training and 296 highly active comparison groups. So in a resistance training program or aerobic training program. We're doing all the genomics paradigm assays. But I just wanted to show one point out. This is a rat study. So male and female rats underwent aerobic exercise program. And we're looking here at lipid regulatory responses, lipids in the blood, acylcarnitines, okay, metabolites. The males are blue. The females are in pink or purple. And red is a positive response and blue is a negative response. This is a heat map. What these are metabolites, and these are actually genes that control lipid metabolism. And you can see there's a dichotomy between the female response and the male response. It's dramatic, okay? I'm telling you today, if women have the same response as men, they do it differently. And we need to understand that if we're going to be targeting therapy to both groups of people. Okay, what are the molecular mechanisms? So this work I'm going to show you was led by Chris Van, who's a postdoc, actually in my wife's lab, but works with us on the Molecular Stride project. And I'm going to show you some, I know I have only 14 more minutes. I'm going to show you some data that at the end of the day relate epigenetic responses or levels at the gene regulation response to gene expression. So we have muscle. This is all in muscle. DNA methylation is a epigenetic mechanism that results in methylation of certain, well, you can have chromosome structural remodeling, epigenetic mechanism. You can have non-coding RNAs as a genetic mechanism, or you can have histone modifications. And the histone modifications are done by methylating sites on the histone, and it compresses that part of the DNA and doesn't make it accessible for transcription into RNA. Okay, I'm only going to talk about that epigenetic mechanism, and that's what explains here. And then I'm going to talk about, and this is how the work was done, and then I'm going to talk about transcriptomics real quickly. So people underwent exercise training, you know that. We took a biopsy before and a biopsy after, isolated RNA and DNA, subjected them to chip technology so we could get a gene expression quantitative assessment for up to 20,000 different transcripts. And we can get, in the infinity array, 850,000 sites in DNA. And then we did differential analysis and pathway analysis, okay? And we did that with this program called Mergeomics, and the whole point of this is we're relating gene to gene, mapping the methylation responses to the transcriptomic responses, right? If you're putting a methyl group at this gene, is that having an effect on the transcript? We can measure both in the same sample. That's the beauty of this effect. Okay, effects on exercise amount and intensity on methylation and gene expression, and the two together on the methylation responses. Okay, I'm going to show you the three groups: low amount, moderate intensity, methylation. These are methylation tags. Is it up or down, and to what extent in response to each group? Okay, and these are volcano plots. I won't explain them here, and I'm not going to even talk about them subsequently. And these are the pathways involved, okay? But this is the core diagram, which is what I want to show you. And this is the sites, and these are the pathways, okay? And it's relating, the change here is related to the change here. So this site in DNA is related to primary biosynthesis. This is nucleotide excision repair. This is sphingolipid metabolism. It's a very ordered response in this moderate intensity, low amount group. Very ordered, right? I think it's ordered, but each site is targeting one pathway. Okay, moderate vigorous. Okay, something's going on here. There's a little crosstalk here that wasn't going on before. Okay, low amount vigorous. High amount vigorous is a little more messy or a little bit more busy. The question is the vigorous busy good, or is it confused? Is the muscle confused? Is it doing too many things at the same time? And is that why we see this contrary effect of vigorous intensity? Okay, now I can pull all three together and you can see the progression across the groups. Okay? Now, gene expression. Okay, same core diagrams, same thing. Okay, here's moderate intensity, very ordered response. low amount vigorous, a little more chaotic, a lot more sites affecting more pathways, right? Doing a lot of things, and high vigorous. Is this good or bad? I don't know. This is what we're about to try to figure out, okay? But it's clear the groups are different in their metabolic response, in their regulatory responses. Each one is different than the other. So you add more amount, you get more of this. You add vigorous to moderate, you get the intermediate response. But it turns out when you look at the specific sites, and they don't kind of line up, right? So what we turn to is what we call the rhythm diagram, the ribbon diagrams. And that's to relate the two together, okay? So let me show you an example. This is the low moderate. So this is the differentiated regulated sites for DNA methylation. These are the differential expressions of the transcripts. So what this does is allows you to say, okay, this DNA methylation response is related to this transcript, okay? And that results in a change in notch, okay, the pathway. And you can see there's a lot of crosstalk here that isn't necessarily related to here, but this is the ordered response that you saw on the chord plots, right? You have a whole group here that are related to the proteasome. You have a whole group that are olfactory to conduction, transduction. You know what that is? Those are G protein transmembrane receptors. That's what those are. Beta receptors, all the stuff like Lefkoid studies. It's just they put it in that named group. We're not looking at smell here. All right, so I put all three of these together for comparison. The groups are different. I'll let you pause and look at that. They're doing it differently. What I've charged Chris to do, and you can imagine this is the next question, is are women different than men in these groups, right? We haven't done that. This is still this is like hot off the press in the last couple of months, but this is, you can see where our work is going here. And then finally, in the last few minutes, the paper that you guys read, okay, the medical students read, right? Okay. So I'll go through it. You've read it already, but you never heard me talk about it. So I'll talk about it. Did you have to present to your whole class? Small groups. Okay, you presented to your small group. Okay, great. All right, so this is Monica Hubel. I've referred to her before. And the bottom line is this is just a cartoon of a cell with a mitochondria and calcium storage, et cetera, et cetera. And there's a whole bunch of action going on here. And the question is, can we identify a target for insulin sensitivity response? Sorry. Can we identify a target and a drug to repurpose here? And so this is Constantine Oliferis. This is CC Ma. They do the causal analysis, right? And Gordon Broderick is an engineer by training. And his postdoc, Matt Morse, did some work on this study. But the thing about Gordon is he designs a circuit diagram, action diagram, that is manipulable. So what he does is he designs these pathway diagrams, and he says, what would happen if I downregulated this protein? What would be the response in the network? So that helps you, and it's done computationally, right? And it allows you to say, okay, my next experiment is actually to go ahead and knock down that protein and see if it does what we presented it to do. That's ultimately the proof. Whether you do it in cell culture or whether you do it in animals, et cetera, you need to go one more step to actually seal the deal. But we have GWAS data, which we aren't using in this study. We have epigenetics, which I talked about. We have the gene expression, we have the metabolism, and we have the phenotype. And then we do this causal analysis and we build a network to identify potential targets. And more linearly, again, we have the people, we have the muscle, we have the arrays. And then what I'm going to talk about is we look at the effect of amount and intensity and sex-related effects on transcription factors. And they say, what transcription targets do these transcription factors target? Okay, so these are the master regulators. What do they regulate? And then we do amount, intensity, and insulin sensitivity related genes. And we put these together and we get causal genes, to simplify it. And then we put these causal genes into a pathway and we say, how do they relate? So here's the transcription factors. Okay, let me give it a thing. All right. These are the transcription factors in female that are related to insulin sensitivity response in muscle. These are the transcription factors in men that are related to insulin sensitivity response. In women, amount is the major player. And they're all negative regulators. In men, intensity is the player. And they're all positive related. Not all, but predominantly positive. If women do the same as men, they do it differently through different mechanisms. Okay, so then we had insulin sensitivity related genes. We had exercise transcription factor genes. We had exercise related genes. And the interplay was 13. So we have the ringleaders for exercise response, the ringleaders for insulin sensitivity response, and the influencers, the transcription factors, all affecting these 13 genes. And these were the genes. All are genes except two. There are two compounds in here. One is beta estradiol, otherwise called estrogen. And the other is tetraphenyl phorbol acetate. We call it phorbol ester. Okay. So who studied with Lori Goodyear? You know that PKC signaling, which is downstream of phorbol esters, or phorbol ester is an analog of PKC, right? Is a big player in insulin sensitivity, right? So, oh my goodness, we're seeing things we expect. VEGF regulates capillary density in muscle or capillary growth in response to exercise training. TGF-beta is a player in cancer regulation, and MYC. So there's nothing here that was surprising. But you put them all into a pathway, and you get this. Okay? So these are our compounds, and these are our genes in that list. And there's a lot of interplay between the two. I mean, between all of them. But something got my attention here. Number one, as I mentioned, TPA is a compound. Question PKC signaling is important. And other beta estradiol. And the other thing that caught my attention is I had published a paper with Shimi Timmons talking about drug repurposing using data from STRIDE PD, not STRIDE 1, and some other studies to show that you actually, EGFR is in fact a target that has a drug, multiple drugs that actually target it, that could be used potentially to study for exercise mimetic effects. So that was a different cohort, STRIDE PD. This is STRIDE 1. Two different cohorts identify the same target. So this is what I just talked about. This is Akshay Barija, who's also, he came to me as a postdoc in muscle, and then worked with Jim White for a while. And now I've drawn him back as actually an analyst. He actually teaches R at Duke to undergraduates, et cetera. And so I'm getting his skills to work on our projects. So it takes a village. This was STRIDE 1, the team. Joe Homard's been a colleague of mine for years on these. And I mentioned Eric, Dustin, and Monica. But these were my crew way back when. There's Joe. And this is all the people that are now working in some way, shape, or form on the molecular STRIDE project. Either collected the samples, et cetera, when we were doing the studies, or involved actually in the analyses now. So I have time for 0 questions. I'm over by 2 minutes. But thank you for your attention. I shared a lot with you today, but I'm happy to answer anything now or by email or otherwise.
A few minutes for questions. Any questions?
Thank you for the talk. It was excellent. I had a question actually about that last paper that's in pre-publication.
Yeah.
I noticed during the section talking about the Markov boundary and the linear regression model that was used to evaluate those. In all 4 models, there was this gene RAS-GRP that was identified. And I really wasn't sure what the significance of that gene was related to the causal modeling.
Well, it ended up being in the larger, analysis, but didn't end up in the Venn diagram, right? So it was, I think that was the insulin sensitivity, exercise insulin sensitivity group, but it didn't get down to the final 13. It was in one of the other circles. Yeah. Oh, come on. I've had a hard day. How about you guys?
Thank you so much for coming to speak to us. Really interesting talk. I'm wondering if you can speak to maybe how you balance as a clinician. The goal of your research is to come up with molecular targets. And at the beginning of your talk, you spoke about can we prescribe exercise? And can you talk to sort of the trade-off of if you were able to prescribe exercise and the additional costs that's associated with that versus being able to prescribe a pill, and how sort of what the trade-offs would be for clinicians in being able to do one of those two things.
Very quick. You want to keep your job or you want?
So a pill does one thing. It's very targeted. Exercise does 100 things. Okay? It affects, but I told you all these things, right? Bone, muscle, cardiac.
I call it positive side effect.
Cognitive function, prevents Alzheimer's disease, just a myriad of things, very complicated, integrated response. I'm talking about targeting a specific disease or abnormality and learning from exercise and how it works to be able to develop a better drug. And not only develop a better drug, but just take one off the shelf that's already there, right? You're never going to get an exercise pill, according to me, because it would have to be a compound of about 10 pills. It'd be this big.
See, he's very diplomatic. I have a question.
Yeah.
That is, I noticed your stride 1, it is surprising to see the insulin sensitivity even not improved as much as the lower intensity exercise, which probably got a lot of, publicity and
No, I got a lot of negative comments from
Oh, really?
my colleagues.
ah,
It can't be true.
okay. But my question is,
Vigorous is better, isn't it?
yeah, my
Right?
question-
More is better, isn't it?
yes.
How can less and
Yes.
less intense be good?
Okay, listen to my question. My question is that it's very known that the cardiac fitness is inversely related to mortality, all cause of mortality.
Yeah.
And in your stride 1 study, your high intensity has better improvement in exercise capacity,
Yes.
right? So the moderate one is in the middle, lower intensity is lower. So which one is better? You look at the parameters of insulin sensitivity, all these things.
Okay, let me,
versus,
I don't have this diagram. I wish I had put it in the pile because this came up in a discussion I had with somebody else. If you take an individual and you put each individual on the line and you graph out or you put a number for the percent change in glucose control, mean blood pressure, waist circumference, HDL cholesterol, triglycerides, and fitness,
yes.
and you order them on fitness, the best fitness to the worst fitness, they don't all line up. There are people that improve their fitness, but not any of the others. There are people that improve some of the others, but not their fitness. So fitness, yes, is a general marker, but it's not all encompassing. My answer is pick out that person, tell me what they want to improve or they have to improve. I'll give you a program that'll probably be best for it, right? HDL is all about amount. So the HDL increased the most in the high vigorous group because they had the greatest energy expenditure.
I changed my mind. Next time I play pickleball, I won't play hard.
There you go. Well, it's good for your insulin sensitivity, but not for your risk, right? You're right. Yes.
You mentioned in the one study that there was a 30% inclusion of minority status. Was there significant results for sub-analysis of that? Did you guys
Too
identify?
small of a sample size.
Too small. For what? And was it 30% for each cohort? For stride 1, 2, 3?
Yes.
And not all of them.
Yeah.
it
Well,
was
we
too
didn't
small?
actually get there. We got to the same as the population, which was about 22.
Stride 5, is that powered to for
Stride
minority
5?
status?
Depends on if NI shall fund it at this stage. Or it'll be administratively withdrawn. Yes, ma'am.
Hi, thanks for coming out to speak to us. I was curious, you mentioned that the sort of definition of diabetes changed
Yes.
sort of since that. Do you think how did that affect your analysis using the data sort of from that pre-definition?
Well, we never had anybody really that high anyway. So we really didn't have anybody in that study that was diabetic. If you go back and look at their fasting glucoses, they were never really that high. But we set that, this was my first study, right? So it's 1998, first human study. That was our best attempt at saying we're not going to have diabetics in this study, okay? Because that creates a question on the part of the reviewer, the IRB, right? So we just said we're not going to have diabetics. That was the upper limit, but they really didn't have a really high fasting glucose anyway. Yes, sir.
Sorry to ask another
Then you've
question
got-
about
then you've got
results that you
colleague over
didn't show. What was the effect of the detraining?
I don't have that much time. So different for different things. The fitness really doesn't go down in 2 weeks. In fact, in some people it goes up. The capillary density is really interesting. Yeah, it went up and then it continued to go up in men. In women at 96 hours, it dropped back to baseline. Yes. Right. The insulin sensitivity dropped down to 96 hours and then rebounded at 2 weeks. That gave us an opportunity to say, okay, what is the chronic effect of this to this, right? Yes, sir.
Thanks for your talk. I enjoyed
Yeah.
it. So there's some evidence that prediabetes phenotype might impact the exercise response for glucose homeostasis. I'm wondering if you considered those phenotypes as moderators or mediators of your outcomes.
Yeah, so our responses in STRIDE PD were less robust in a number of different outcomes. And in particularly 10 years later, we bring them back 10 years later for another assessment. Not a muscle biopsy, but all the other phenotypic stuff. And we saw that actually fitness dropped off more rapidly in the pre-diabetics in 10 years than it did in the non-pre-diabetics, for example.
Did it matter whether they were included because they had high HbA1c or whether they had high fasting glucose or impaired glucose?
Yeah, it was the diabetes prevention program criteria. We wanted to make sure we were actually reproducing that group. So it's 2 fasting glucoses over 95. Come on, give me one more.
Okay.
Oh, way back in the back. Who said you could sit back there?
I'm really curious about pregnant women and with like changes in hormones, and I was wondering if you could sort of postulate about that. I don't think they're included in your study.
Unfortunately, I can't because I haven't studied that, and I haven't studied it enough to actually comment on it. I do know that exercise is good for pregnant women. I don't know whether or not, and if you believe Lori Goodyear's data, exercise women have the offspring of exercising women have better glucose control. Not only when they're infants, but when they grow up.
And to their grandchild.
And catch this, also true for exercising men. Yes, some epigenetic thing gets carried from the sperm to the egg. And we don't know what that is.
All right, thank you so much.
Hopefully I met expectations. Yes. Thanks for having me. Oh, yeah.